So today we're picking up where we stopped yesterday with gene discovery, and we're going to talk about the other major strategy that's been followed in social science genomics, following the example of medical genetics for genome-wide association studies. So we're going to talk first about what GWAS are and how to interpret what comes out of them. And then we're going to go through an example of the GWAS for educational attainment that have been conducted. So in contrast to the candidate gene studies, which were focusing on a single or small set of genetic variants where there was an a priori hypothesis about why there would be an association, GWAS is, in a sense, the complete opposite approach. It's an atheoretical testing of all of the SNPs that you've measured on your SNP array. So these days, that's about 10 million SNPs. And one thing I'm highlighting here on the slide is that even though this is a large number, like 10 million, it's still a lot smaller than the total number of genetic variants in the genome, which is something more like 50 or 60 million. We set a, so let me just say something about, let me pause here on this atheoretical part and the lack of a hypothesis. So I think that's where a lot of social scientists, when they first hear about GWAS, get a little bit puzzled or taken aback or even repulsed, because that sounds like data mining. Which is something that we all are taught not to do. We're supposed to come into empirical work with hypotheses. So to understand why this was appealing to geneticists, remember the situation. In medical genetics, there had been this period of time of coming up with good hypotheses about where to look for a candidate gene based on linkage studies, and then having good biological reasons to look at a particular gene, like in the ApoE example, we're finding the sequence of base pairs that coded for the beta amyloid protein. But then as the field moved to more complex diseases, there weren't such good hypotheses. The linkage studies were underpowered. They didn't know where to look. They didn't, the results that had been found didn't replicate very well. So there was a recognition that we really, for complex diseases, we don't really have good hypotheses about which genes are going to be involved. And maybe by an atheoretical approach, we'll learn something we didn't know about the biology underlying diseases. Yeah?
I have one question. So if it's a theoretical testing of all, why do we use like the half a million to 2.5 million and not just like straight up like a full array of SNPs? Like what's in like this 2.5 million compared to like the broader set of SNPs that you can have to do the GWAS?
You're asking what's missing from the 2.5 million relative to the 50 or 60 million?
Yeah.
So as Andrea talked about, when the SNP chips are designed, there are various considerations about which SNPs you measure. So one thing is you want to have SNPs measured every so often in the genome, so they're not too far apart, but they can't be too close together either, because then they interfere with each other chemically when you try to measure them. And then you try to use information from the reference samples of, say, European ancestry individuals to try to identify the haplotype blocks where there's strong linkage disequilibrium and try to make sure you're covering the different haplotype blocks in the genome. So the goal is to choose these 2.5 million SNPs or 4 million SNPs these days, according to what Andrea told us yesterday, so that you're covering, given the LD structure in the genome, most of the variation across people in the population, but you're not getting every SNP, and so you're going to miss some of the common variation across people, and you're certainly not getting rare SNPs at all. You're not even trying to get very rare SNPs or rare variants with SNP arrays. Okay, So in the social science genomics area, it's a similar motivation of why atheoretical testing might be attractive. We really just never had good hypotheses for where to look for associations. Okay, so an immediate problem with testing so many variants is that we have multiple hypothesis testing. So the solution in GWAS is to adopt the stringent significance threshold of 5 times 10 to the negative 8th, as we've talked about. There's two different justifications for that threshold. There's a frequentist justification, which is that 5 times 10 to the negative 8th is the Bonferroni correction for 1 million independent loci. What do we mean by this 1 million effectively independent loci in Europeans? It's that taking into account the LD structure in the genome, when you test your 2.5 million or your 10 million SNPs, there's correlation across those tests. So the number of independent tests is roughly 1 million. Now, caveat here is if you go to sequencing data, where now you're including rare variants, that's going to be the wrong number, because the rare variants are not in high LD with other variants, with the common variants you've measured. So if you do the equivalent of a GWAS with sequencing data, we would really need a more stringent significance threshold. And there's an estimate that it should be more like, I think, 5 times 10 to the negative 10. Is that right? Do you remember?
I think there's 1 times negative 8 and Daniel et al., but maybe they've updated it. I don't know.
OK, yeah. Well, it's more stringent for if you do sequencing data. OK, so that's the frequentist justification. The Bayesian justification, which we talked about yesterday, was that you need the stringent significance threshold given that you have a low prior for any particular locus, and the prior is coming from the number of effectively independent tests is the fact that you don't really know where in the genome the effect is, and that there's this number of effective loci. So these were also on the problem set going through these justifications. And as Andrea mentioned, there was discussion in the early days of GWAS about what the threshold should be, and over a few years, both of these kinds of arguments converged to this value. OK, so what we'd like to estimate in a GWAS is the model that comes as close as we can to the causal model. But notice that since we can't actually estimate the multivariate regression with all the genetic variants in the genome, which would be the causal model, because we haven't measured all of them. So here we're just indexing up to k, the k SNPs that we've actually measured in the data. Notice also I've labeled this coefficient beta SNP rather than beta, because it's not necessarily the same as the causal beta. So why is it not necessarily the same as the causal beta? Yeah.
It might be a SNP that is in LD with the actual causal one.
The SNP that's in LD, and what about that other SNP? How come it's picking up? Why would it pick up the effect of a SNP it's in LD with? Why isn't it just, can't you just control for that other SNP?
So I think a lot of times they only measure one of them on the SNP chip, so you wouldn't actually have the causal one measured, or they would just be inherited in tandem, and so you wouldn't be able to exactly know, or you wouldn't have any variation to parse out which one it is.
So let's postpone talking about that second issue that you just raised, but the first one was exactly right, exactly right here. So the idea is you have a SNP that you've measured, and then there may be another genetic variant that you haven't measured, and maybe it's the variant you haven't measured that's causal, but if it's in LD with a SNP that's on the SNP array, it's going to be picked up by the beta for that SNP. if you could estimate the true causal model and control for that other variant, then the beta, would be 0 if the SNP actually doesn't have a causal effect itself. But because you can't control for that other variant, the beta is going to pick up the signal from variants that it's correlated with. Okay. All right. So we'd like to estimate this model. Well, we're going to immediately run into a problem. What problem might we run into if we try to estimate this regression with 10 million SNPs? Multicollinearity is one problem. That's exactly right. It's actually, I want to focus on a different problem, but you're right. That would be a problem because there might be very strong LD between various SNPs. In general, there will be, and that's a problem. What else? Yeah.
Degrees of freedom.
And say more about degrees of freedom.
That we have more SNPs than we have sample size.
Then we have what?
Then we have participants in our sample.
Exactly. More SNPs than people.
It's also multicollinearity, right?
OK. There's a sense in which that's also multicollinearity. Fair enough. So I'd call it the dimensionality problem. You try to run this regression. It can't actually be run. You try to do it on Stata, and Stata is going to give you an error message unless you have more people than you have parameters, which is the number of SNPs plus the number of control variables. Which still there's never been, as far as I'm aware, there's never been a GWAS run with 10 million people. OK, so the standard solution in GWAS is to estimate univariate regressions, to go SNP by SNP, and the same set of controls, but now just one SNP at a time. And now I've labeled this beta something else, beta GWAS, because that's yet again something different from the causal effect or the beta SNP. OK, so we'll dig into what this beta GWAS is in a moment. But one thing that might be on some of your minds, if you've done big data analysis before, is, well, we have a solution. They have a class of solutions for dealing with estimating models where you have more regressors than people, and that's in the machine learning set of approaches. So why haven't those been used? And I think to date, so there has been some exploration in this direction, but I think there's been two main reasons why it hasn't been very common. One is that, at least until very recently, the well-powered GWAS have been the result of doing meta-analysis across many samples. That's how you got big enough sample sizes to be able to do GWAS that was reasonably powered. And it's very hard to, you can't get access usually to the individual level data to literally pool the data across data sets because of IRB and privacy restrictions. So you have to run GWAS in each of the data sets and then meta-analyze. And the machine learning methods don't work when you're doing that. They work on when you have a data set of individual level data. The other issue is computational constraints. When you have, they may work in principle when you have 10 million regressors, but in practice, that's an enormous computational burden when you have 10 million regressors and say 1 million people. Okay, so I think there are reasons why we might expect to see some more machine learning approaches in the coming years because these barriers are lifting. There's now increasingly large samples where you do have access to individual level data, like the UK Biobank, and there's cloud computing solutions that allow you to do things securely and on a large scale. But this hasn't been explored very much yet. OK, yeah.
So John was asking, can't you just run the regressions one chromosome at a time? Like, so you could take the UK Biobank, and if there's fewer than 500,000 people on SNPs on a chromosome, you could do an OLS regression on a single chromosome, right?
Yeah. So that's right. and I think if you did want to pursue the machine learning, that's probably what you would do, is run it chromosome by chromosome. Doing the OLS regression would still be problematic because of the multicollinearity problem, even if you have enough people to do it, to do the OLS regression on each chromosome. So there actually is a method called conditional joint analysis that was developed to try to approximate what you would get from running the OLS regression to deal with the multicollinearity problem, and it's a stepwise regression procedure. Basically adds SNPs as long as it doesn't cause problems with the multicollinearity, very roughly. And it works reasonably well, and it is widely used, but it's not the standard first thing that you do with a GWAS. The standard thing is these SNP by SNP OLS regressions. Then often you see them supplemented by people applying the conditional joint analysis, which ends up adding a relatively small number of SNPs and is restricted to SNPs local that are in LD with it on the same chromosome. Okay, so I want to dig into a little bit into interpreting the GWAS coefficients. So remember, here's the causal model that includes all J variants in the genome. Here's what we're estimating, the SNP by SNP regression. So as we mentioned, the beta that you get from the GWAS is not the same as the causal effect estimate or the estimate from the full model. And in particular, this is going back to what we were saying before. You're going to pick up the effects of SNPs that you're in LD with. What we talked about before was being in LD with SNPs that you haven't measured. Here, we're also going to pick up the effects of SNPs that are measured, because we're just not including them as control variables in the regression. Okay, so you should think of the GWAS coefficient as the true causal effect of the SNP plus the effects of other SNPs that it's in LD with that may or may not have been measured. Here's a picture that Patrick created at some point to illustrate this idea. This is what this is showing on the left is the true causal effects of 5 SNPs that are close to each other on the genome. The y-axis is the size of the effect, and these are 5 different SNPs. And then on the right, we're imagining that we do the GWAS now for SNP number 1, and SNPs 2 through 5 are not in the regression. And so the effect we're estimating for SNP1 is bigger than the true causal effect in this example. It could be, in practice, could be bigger or smaller. But here it's bigger, and it's picking up some of the effect of SNP number 2, some of the effect of SNP number 3, which is the white part, I guess, is what is being picked up, and none of the effect of SNP number 5. It might be picking up none of the effect of SNP number 5, either because SNP number 5 is not, well, I guess it would be picking up none of SNP number 5 if SNP number 5 is not an LD with SNP number 1. So in general, the GWAS coefficient is going to be the sum of these causal effects weighted by their correlation with the GWAS SNP. Okay? All right. So because of this, when we run a GWAS, if there's a true causal effect somewhere in the genome, what we typically see is not just that true causal SNP associated with the phenotype, but also a whole region of SNPs nearby that are in LD with that causal SNP. So it's we refer to that often as a genome-wide significant locus of SNPs. For accounting purposes, we don't want to count all of those as hits because we think usually there's just one or a small number of true causal SNPs when we have a whole locus of SNPs that are associated. So we pick one of them to be the representative for that locus. And there's done in different ways, but often it's the SNP with the smallest p-value in that locus. And we also try to use an algorithm such that the set of lead SNPs collectively are approximately uncorrelated with each other, because we don't want to be double counting a locus when we're counting up the lead SNPs. COJO, like I mentioned before, this conditional joint analysis can do a bit better because it can identify cases where there might be more than one causal SNP within a locus, which and here, if we make sure that all the SNPs are uncorrelated, we're going to potentially be missing cases like that because we're just going to pick one representative from the locus. So there are methods like COJO that try to do a bit better than this. OK, so we talked last time about the three big problems of gene discovery. How does GWAS do at addressing these problems? Well, for multiple hypothesis testing, we are transparent about testing all of the measured SNPs. And we use the genome-wide significance threshold to deal with that issue of the testing all the SNPs. What we don't deal with in a typical GWAS is potential multiple testing across phenotypes. Now, traditionally, or the way GWAS has been done for a long time, that hasn't been much of a problem because it was such a big effort to do a GWAS meta-analysis and getting lots of cohorts to do the analysis and upload results, and it took several years that you weren't going to test multiple phenotypes. You were focused on one phenotype. That was the project, and you're going to publish the paper in the end. I think recently, with the availability of large data sets like the UK Biobank, this may reemerge as an issue. So something to potentially be careful about. Okay, the second issue is gene-environment correlation. So one of the big types of gene-environment correlation, and the one that's gotten the most attention until very recently in the genetics literature, is population stratification. And GWAS has built into it, or the GWAS data makes it pretty simple to take an approach to dealing with population stratification. And that is to use the genome-wide data itself to estimate principal components, and then control for those principal components. So I'm not going to talk much about this because Andrea showed you those nice pictures of how well estimating the PCs can separate people geographically, and that's the idea. You kind of separate out people of different ancestral groups, or they're not really groups, it's a continuum on the principal components, but you capture ancestry of the principal components and control for it. So what we now know, so this for a long time, this was thought to pretty much fully deal with the problem of population stratification. In the last few years, it's become clearer that it's not a complete solution to the problem, and particularly, as GWAS have gotten larger, we've been able to detect more of the problems with the population stratification, with the PC approach. But I'm going to leave that to Patrick to talk about later today. One thing to mention is that the way you could fully deal with gene-environment correlation is to conduct your GWAS within families, or control for parental genotypes, or almost as good would be the sibling GWAS with fixed effects. The reason that hasn't been done is because of sample size constraints. It's just there aren't as big family samples. But that's starting to change, and there's a paper that some of us contributed to that is now on bioRxiv, should be published soon, that does some within-family GWAS for a number of phenotypes. Still, the power is relatively low relative to what we can do with GWAS with with unrelated individuals. But I expect this to become increasingly important in the coming years. And that would be a complete solution to the population stratification problem, as well as other forms of gene-environment correlation. Yeah?
Feel free to answer this question, but I'm curious, why don't we use something like variable education, like something to control for SES?
So the question is, why not include more control variables in the GWAS to try to deal with some of the issues of gene-environment correlation? And I think, in principle, that's a good idea. The way that GWAS had been done early on, it was very hard to include more than just basic controls like age and sex, because, in principal components, because you had to coordinate this analysis across many data sets, like 50 or 70 data sets, and they would have different measures in those data sets, so they might not all have parental income, in fact, or parental SES. In fact, very few of them would if they were medical genetics cohorts. I think it's a great question for, a great idea, potentially, for data sets like the UK Biobank. Actually, though, the UK Biobank does have measures that you could link with parental SES, like where your family grew up. So I think that would be a reasonable approach to try to deal with some of the gene-environment correlation if you have a large enough data set with those measures. Yeah?
Going back up to your full hypothesis testing, the point about having like many large-scale GWAS. Given you're saying we're seeing GWAS for a bunch of different phenotypes, and so now maybe our type 1 error rate is no longer controlled because we're looking at a million different phenotypes. However, given what you were talking about before with the post, I forget what the vocabulary is, but like you said, what's the probability of it actually being a hit conditional and being significant is like incredibly high when we have a 10 to the minus 8 p-value. So does that mean that if we're using stringent p-value thresholds, like type 1 error may go up, the false discovery rate may not be as bad, or I'm just trying to think of how that would link. Does that question make sense, what I'm asking? Like.
Yes. So the question is it still the case, that from a Bayesian perspective, a hit is very likely to be true based on the kinds of calculations we did before, even if there's unreported tests. And I think the answer is yes. I need to think about it. Let me think about it a little more and get back to you later. Okay, so then low power. So this in the early days of GWAS, the power was exacerbated by the idea that now we're going to adopt this really stringent significance threshold. And so it became clear immediately that if GWAS was going to be a research strategy, you couldn't just, none of the medical data sets were large enough to actually do a well-powered GWAS by themselves. They were of 5 to 10,000 people, and that wasn't going to be well-powered. So the concern about power led to a new infrastructure for doing this kind of research, which was creating consortia, these cooperative enterprises among data sets where they would work together and each do a GWAS. There would be a pre-analysis plan that would be circulated to everyone to coordinate the analysis, and everyone would upload results to a centralized, secure server, and then you would meta-analyze the results. And so it led to a complete change, actually, in the culture of research in medical genetics, where previously the data sets were very possessive about their data, didn't want anyone else to analyze their data because it was their resource. And then once the GWAS era began and it was clear you needed to cooperate, then every data set was contributing to every GWAS, and they were all working together. Okay. So there have now been GWAS for many, many, many phenotypes. I've listed just a few of those that we might be most interested in on this slide. And I've listed the most recent papers. I've tried to list the most recent papers for each of these phenotypes. In medical genetics, height and as well as BMI, but particularly height, have been kind of model phenotypes. Not so much because people care that much about the genetics of height, but more because it's a highly heritable trait, and it's been available in the largest samples for doing GWAS. So we've learned a lot about the methodology and about the kinds of things we can learn from GWAS by doing GWAS of height. Okay. So so now when you have a GWAS paper, the first step is identifying these lead SNPs. And then typically these papers include lots of other kinds of analysis. So a lot of that work in medical genetics papers is follow-up work to try to identify the causal SNPs or the causal genes. Typically that work is focused on bioinformatics that uses these other databases about gene expression and EQTLs, the kinds of things that Andrea talked about. In some cases, there's wet lab follow-up where you try to do experiments to manipulate a gene and knock out a gene in an animal model and confirm that which is the causal one. There's also analyses to look at how much the SNPs that are identified in the GWAS are expressed across different kinds of tissues or cell types to understand how it might work biologically. GWAS results can be combined with GWAS results on other phenotypes to calculate genetic correlations. And that's something that Raymond is going to be talking about. And then you can use the GWAS results to create weights for constructing a polygenic index, which you could then apply in other data sets that have genome-wide data. And that's what Patrick's going to be talking about later today. So before we move on from talking about the methodology, I just want to mention one last thing about candidate gene studies, which is that I want to be clear that although they had lots of problems in the way that they were done, there's nothing wrong with candidate gene studies in principle. And in fact, they are often used in other contexts successfully where there's reason to look at particular SNPs or particular genes. So to deal with the problems that came up with multiple hypothesis testing, you could pre-register your analysis and use the appropriate significance threshold. You could deal with population stratification. If you have genome-wide data, you can control for the principal components, or you can conduct the analysis within a family. And you could use large enough samples to not have low power. So it's not like candidate gene studies are always going to be a problem, but the way they've been conducted in social science has been problematic. One way to do them better is to, instead of relying on hypotheses that are pretty flimsy, you could choose empirical candidates, by which I mean GWAS hits from related phenotypes, and then study how well they're associated with some other phenotype that you're interested in. OK. So any questions about the GWAS approach before we talk about the example of educational attainment? Yeah?
Sorry, just to go back to the regressions that you're showing. So it strikes me as a classic issue of limited variable bias.
Yep.
I was wondering if there's any scope for some of the usual fixes we have for limited variable bias, IVs or something like that. I know you can't really plausibly think of an instrument that just shifts one SNP. Is there anything like that we can do to address it?
So the question is whether there's any kind of approach to omitted variable bias, like instrumental variables, that you could use to deal with the problem.
Get more to the causal beta than just some beta that's, the real beta plus a whole lot of bias, the way I see it.
Yeah. So to try to get at the causal, to get which is the causal variant. I can't think of anything to do, but
I mean,
Yeah. Okay.
I have a question.
Yeah.
So I'm just curious, because so far we use analysis kind of like some of the problems I'm trying to do gene discovery. But let's say I was less concerned about doing gene discovery and really just want to estimate a polygenic risk score for like a sample of 500 people. Do I still have to be concerned with power in those scenarios if my goal is not gene discovery?
The question is, do I need to be concerned about power if my goal is not gene discovery, but constructing a polygenic index? Well, the answer is yes. In brief, the reason is that in order to get the right polygenic index, to get a predictive polygenic index, you need to get good estimates of the weights to use on the SNPs. And if you have a small sample, you're going to get terrible estimates of the weights. They're going to be extremely noisy, and they're not going to be a good predictor. Patrick is going to talk about that in more detail. Any other questions before we go on to educational attainment? Okay. Okay, so as I mentioned, a number of us who are instructors are affiliated with the Social Science Genetic Association Consortium. This is a consortium that David and I and Phil Kohlinger founded in 2011. And it was the original motivation for its formation was that we were doing this work on candidate genes and seeing that it wasn't replicating, and we were in the midst of doing this work, then looking at candidate genes for cognitive performance and documenting that a lot of those didn't replicate very well. And we were trying to think about how we could proceed with doing social science genetics in a more productive way. And it was clear that the medical genetics community was, had good reasons for moving to this GWAS approach. So we wanted to, we thought that made sense to us, and we wanted to try to adopt the same kind of approach for phenotypes of interest to social scientists. So to be, it was not obvious at that time that this was the right approach. In medical genetics, even though the first GWAS had been done in the mid-2000s and there were some successes, by 2011 there had been GWAS of many phenotypes, and many of them, they were kind of the typical sample sizes were still in the range of like 30,000 to 50,000 people, and they were looking at increasingly complex phenotypes like depression and schizophrenia, and it was pretty typical to find absolutely nothing from the GWAS, 0 hits or maybe 1 hit, and so a lot of people looked at that and said, this is a reflection of flaws with the methodology. The GWAS is not going to work. And there were different reasons people had in mind for maybe why it wasn't going to work. Maybe there was too much heterogeneity across the cohorts. So this whole idea of meta-analysis, you were adding too much noise by averaging over different environments. You were adding more noise than you were going to solve by increasing the sample. So it was not clear that this was going to succeed, but we thought it made sense based on the statistical soundness of the approach. We were confident it was going to work. The first question we had to face was what phenotype to study, and the conventional wisdom at the time was that we should be focused on phenotypes that were as close as possible to the kinds of phenotypes that where things had worked in medical genetics, like the diseases where it's close to the biology and you can get good measures of it. So the argument was we should focus on things like well, cognitive performance maybe, but even better would be like processing speed. How fast you can click a key on a keyboard, because that's very close to the biology, and so presumably is more heritable, and there's a better chance that there's some direct biological connection that we could discover through GWAS. The problem with studying that kind of phenotype is that it's not measured in many of these data sets. There weren't, medical genetics data sets were not focused on things like how fast you could click a key on a keyboard. So we were thinking, as an alternative, you could try to study a phenotype that was available in a very large sample that would be worse on these other dimensions, less consistent across cohorts, less heritable probably, less biologically proximal. So the prime candidate that we stumbled on was educational attainment, and that's because it was measured as a background variable in many of these medical genetics studies, because they wanted to control for it. So we did power calculations, and this is kind of an ugly picture, but this is literally the Excel figure we put together when we were trying to make this decision about what to study. What we did was, so these are power calculations. The x-axis here is the sample size for the GWAS, going from 0 to 100,000, which was our aspiration. We thought maybe we could get to 100,000. The y-axis here was r squared. Notice that it goes from 1/10 of a percentage point to 1%. People thought, this is much smaller than the candidate gene studies were claiming, and we thought this was, we were being pretty conservative by choosing this, when in fact what we learned actually was that the effect sizes are 10 times smaller than this. And then we were doing calculations for different levels of retest reliability, so kind of amount of measurement error in the phenotype. So we thought of like a rho equals 1, this green would be like a really well-measured phenotype. And what this is showing is isopower curves. So we're looking at, for 50% power, what's the combination of sample size and effect size that gives you 50% power for a well-measured phenotype. So we thought of this 1 or 0.8 as maybe corresponding to cognitive performance, and we thought we would need to do a GWAS in 35,000 or 40,000 people, if this is the effect size here, this 0.1%, to have 50% power, which was what we thought would be reasonable to aim for. That was impossible. Combining, we had done this meta-analysis with GWAS data from the candidate gene replication paper. We were able to put together a sample size of 10,000, and we didn't think there was much more data out there than that. The alternative would be educational attainment, which would be much worse measured. We were thinking it would be like a rho of 0.4 or so. And there, to get 50% power at this effect size, we would need a sample of about 80,000 people. That seemed to us much more doable. So it was this that convinced us to pursue educational attainment. So this would be our proof of concept for other behavioral phenotypes. And the result of this effort was a paper we published in 2013. And we did ultimately end up getting a sample size of about 100,000 by meta-analyzing 41 data sets. This goes back to the question before about control variables. We followed the convention in medical genetics of having a minimal set of controls, just age, sex, and genome-wide principal components. There really wasn't much available in these, most of these 41 cohorts that we could have controlled for that would go beyond that. And what we found was 3 genome-wide significant hits. So this is a Manhattan plot. Let me walk, I know Andrea walked through it, but let me go through it again, because I think it's important to know how to read one of these plots, and repetition might be helpful. So the x-axis here is location in the genome. divided by chromosome. So this is chromosome 1, 2, 3, and so on, up to 22. So this includes only the autosomes. It's rare to include the sex chromosomes in GWAS, although in the more recent EA GWAS we have included the X chromosome. The Y chromosome is almost never analyzed for reasons I don't fully understand, but I think there are some technical challenges. The y-axis here is the p-value on a negative log 10 scale. So that means that higher numbers are more significant. And this dark line here corresponds to the genome-wide significance threshold. There are some, so there are on this graph, a million some points, but you can't see most of them because they're lying on top of each other down here in the sub-significant area. There are some gaps. Those are things like centromeres, where it's very hard to measure the SNPs. You see towers. So this is a bit of review, but why are there towers in these plots? LD, exactly. So this is what we said before, that you get a whole locus of associated SNPs because the nearby SNPs are in LD with each other. And that's why these are called Manhattan plots, because of these supposed to look like the skyline of Manhattan if you actually have some hits. And what we found in this first GWAS was 3 genome-wide significant loci that you can see here on chromosome 1, 2, and 6. And then we had a replication sample. So after we did this analysis, we then recruited additional cohorts and got 25,000 people from 12 data sets. And we could adopt a less stringent significance threshold. We just Bonferroni corrected for 3 SNPs. But at the 0.05 divided by 3 threshold, all 3 SNPs replicated. So we viewed that as big success. We showed that GWAS worked. We also had the first credible estimates of effect sizes that we could expect for behavioral phenotypes. They were really small. It was like the 0.02% R squared for each of these hits. And that could form the basis for power calculations for future studies. As I mentioned last time, the general reaction to this paper was not positive. People said, you see, GWAS does not work. You find basically nothing, and it's a waste of money. The least important results science ever published. So we had a different view. Our view was that the small number of hits we found was a reflection of lack of power. That we still had too small of a sample size to see very much. This was something that was similarly the advocates of GWAS and medical genetics were pushing this view. And it was gradually the case in medical genetics as GWAS were done in larger samples, you stopped seeing these GWAS with no hits, and you started to see more and more hits, and the medical genetics community was becoming more convinced that GWAS was successful for complex phenotypes. So we increased the sample size. We immediately upon publication, we embarked on increasing the sample size for EA. We published a few years later EA2. So now we got the sample size up to 300,000 individuals in 63 data sets. So the samples, because of the falling cost of genotyping, there was just dramatic growth in the number of cohorts that were out there with genome-wide data, and existing cohorts were adding participants, genotyping more people. So so the sample size grew a lot. And when we analyze this data, we now have 74 lead SNPs. They're marked with the orange dots. And then I've also marked on this Manhattan plot with X's the 3 SNPs that were identified in the EA1. So they continue to be significant in the larger sample. During the process of revising this paper, the first release of the UK Biobank came out. The UK Biobank has 500,000 people, but it actually came out in 2 stages, and the first release was 150,000 people. Of those, there were 110,000, 111,000 with EA measured, and they were of European ancestry and passed the quality control. And we used those as a replication sample in this GWAS. And the replication record was quite good. So not literally the case that every SNP replicated at Bonferroni correction, but what we could do was calculate what replication record you would expect if the 74 hits were all true hits, and you take into account that there's a certain amount of power. We have 111,000 people in a replication sample. What would we expect to see when we try to replicate? And the replication record matched up pretty well with what we expected. Patrick's going to talk, I think, a bit about how you do those kinds of calculations. Yeah.
So, because the dots are quite small, but it looks like there's one orange dot for 1 power. But sometimes I see 2 dots in 1 power.
Yes.
So then I don't really, because it's like the definition of orange dot is kind of
Yes.
set, right? So then why
Yes.
there's more than 1 dot?
Good question. So why does it look like in some places like here, there's 2 orange dots that look like they're on top of each other, and even though they're supposed to be uncorrelated SNPs? And the reason is because the genome is actually very big, and we're packing it into a small space here for the graph. So they actually are not on top of each other. They just look on top of each other in the figure.
Very tiny towers.
Yeah, I mean, it could also be that they are actually close to each other, but that the LD block, there's an LD block on one side and LD block on the other side. Yeah.
I have a question. So like most of this is done with European ancestry individuals. But
This is all European ancestry
but
individuals.
and it has to do with the fact that there's like more data on that, right? So we would have like data for like different ancestry groups. We would do like the same, or how would that like? Would it be like do like separate models for different ancestry?
Yeah, so the question is, how would you incorporate other ancestry groups if you wanted to do them in the same GWAS? And that's a question of ongoing study. I think there is no accepted way of doing it. I think the current approaches are to do separate GWAS in the different ancestry groups, which means that in the other, you might have a very large European ancestry sample and then a very small African ancestry sample, and so you just don't find very much in the, African ancestry sample. I don't know, Patrick, do you want to say anything about this?
Yeah, no, I mean, I think that's just what Dan said. I think the standard thing is to just separate your data into homogeneous groups. And there's a few projects, like the 10 UKB project, looks at the like 6 different groups within the UK Biobank, but and then some of these GWASs have like 5,000 people in them, and so it's not very current in how much you can learn. But the data are growing. There's big efforts by a lot of groups trying to gather more data that are more representative of the global population. There's other methods that are being worked on that maybe allow you to incorporate data in different ways. Elizabeth's going to talk more about those kinds of things next week.
Yeah.
Sorry, just to follow up on that a little bit. So just from an econometric standpoint, it just seems to me like adding different groups where the variation in the genes is different should kind of help identification in a way. Am I thinking of it?
You're absolutely right. So the question the question was, it would seem like having different ancestry groups, and particularly different groups with different LD structures, should give you leverage for identifying the causal SNP. And that's exactly right. That's the key insight underlying fine mapping methods, a number of fine mapping methods that try to use data from different ancestry groups and combine them to identify causal SNPs. You still need well-powered GWAS in the other groups in order for those to be successful. So I think there have been examples of successful use of that approach to identify causal SNPs. But I think it would still be, I think the power would still be too low to use it for something like educational attainment in other groups.
And there's getting to be quite a bit of data in East Asian ancestry samples. So the China Missouri Biobank and Biobank Japan. And so, you know, neither of them have really great measures of educational attainment, but they do have measures for other phenotypes.
Yep. Yeah, that's a good idea. Yeah.
Spencer, so then why wouldn't you always like just pull your samples and use as much variation as you can? Why are you doing it on different populations for the GWAS?
Oh, so why not pool the different ancestry groups together?
Yeah.
Patrick, do you want to answer this question?
I mean, it's a population stratification issue, right? And so there are a lot of variants that are just more common in East Asian ancestry populations. But also a person who has East Asian ancestry probably grew up in very different environments. And so you have ancestry then as an omitted variable. And so if you pool everyone all together, then you have really bad bias due to population stratification. I mean, like there are people who are working on. I mean, like I have a project even called MAMA that's designed to try to take separate GWAS and say, well, how much can you learn by pooling them in a way without introducing bias? So I think there is information there. People are working on those kinds of things. But yeah, just kind of throwing them all into one big pot and doing a GWAS though is like that's not going to work because of the stratification between the populations.
Yeah. So you certainly couldn't just pool the data, you analyze the data with different groups in the same sample, because then you have the chopsticks effect kind of problem and more subtle versions of it even with PC controls. So what you need to do is separately do the GWAS, and then you still need to somehow model the LD differences and allele frequency differences between the groups if you're going to meta-analyze them, and that's exactly what MAMA and these other methods try to do. Yeah.
So following up with that question of like pooling different cohorts, I was wondering like, so the concern of like working with a smaller sample, I am assuming would be like you will be missing like slightly rare like variants which aren't seen in like samples that are pretty high. But with that caveat, if there are common variants across ancestries, wouldn't pulling them up still identify like common variants that are like common across like ancestries, and still you may be able to increase the sample size?
So the question is, if we just focus on variants that are similarly common across ancestry groups, by pooling the ancestries, could we increase the signal? Maybe. I mean, I don't know. I guess it would depend on the case about whether you're how much bias you're introducing relative to increasing power. But I think if the goal is gene discovery, the method Patrick developed, MAMA, is the optimal way. I mean, is the optimal way to combine across samples, and so you get the boost in power without introducing bias. And if you have similar allele frequency and LD differences, it's going to be all, you're just going to maximize the power increase by using a method like MAMA. Yeah.
Sorry, just one more clarification question. So when you can control for population stratification by extracting principal components within an ancestral cluster, but that doesn't work at a more macro level across ancestral groups?
Well, it would work at a more macro level, but what you would be capturing then is that the top principal components would be capturing differences between the groups, and then you'd be pushing the differences within groups to lower principal components. I'm not sure. It wouldn't deal with the LD differences between the groups. I don't know. Do you want to say more about that?
Yeah, I mean, I think the issues. I mean, population structure is really, really complicated. It's super high dimensional, right? And PCs are linear, And so if phenotypes are stratified in nonlinear ways, then the more diverse your data are, the worse PCs the worse PCs will likely to be at capturing it, right? Because there's just more room for these nonlinearities to just creep in and cause problems. So like it might capture some of the first order effects, but when we're looking at behavioral phenotypes, we're looking at very small effects, and so even a little bit of stratification due to nonlinearities in the phenotypic stratification could add up to be very large in the end relative to what we want to see. So that's why we say, well, let's first restrict to something somewhat homogeneous, such that these linear PCs are a good approximation, within that group.
That's a great explanation. So then you essentially have non-parametric controls by across the samples. Okay. So this was EA2. EA3. So then we collected more data and used a full UK biobank and an expanded and a sample, a pretty large sample from 23andMe, and got up to as well as some other data sets. The Estonian biobank was available by that time, and some others. Got us up to 70 data sets and 1.1 million individuals. So the Manhattan plot now looks entirely different. This is the genome-wide significance threshold here, and you can't even see anything under the genome-wide significance threshold. Just all across the genome, you're seeing genome-wide significant hits. There's 1,271 lead SNPs. Here for replication, rather than having a separate replication sample, we looked at replication of EA2, combined discovery and replication sample in the new data, and vice versa, and continued to see good replication across those subsamples. And then I'm going to, this is a sneak preview about EA4, which is currently under review and hopefully will be published later this year. This is now a discovery sample of 3 million people. So this is entirely from an increased sample size from 23andMe. And I didn't write the number of lead SNPs on the slide. I should have. What is it like?
1,271.
No, that was EA3.
Oh, right.
EA4. It's more than that. It's several thousand. I'll get back to you on the exact number. And then another thing we did in EA4 was took advantage of the large sample size and did a dominance GWAS. So we also did a GWAS of the dominance deviations. So we talked about that before, and I said, well, sample sizes, that you may not want to do that with smaller sample sizes because of the additional parameter. Well, we did it for EA4, and this is the genome-wide significance threshold in the dominance GWAS. Nothing was genome-wide significant, despite having this is not actually quite the full 3 million people. It's like 2.7 million people. Still extremely well-powered. So the fact that we see no genome-wide significant dominance hits means that we can rule out effect sizes in terms of variance explained for these for dominance effects of 1/10 the size. So remember the effect sizes for the additive effects, the largest additive effects are of the order 4/100ths of a percentage point r squared. And for the dominance effects, we can rule out effects of the order 4/1000ths of an effect of a percentage point of r squared. And we can also estimate the overall variance in educational attainment due to dominance variance, and we get a pretty tightly estimated 0. So there's actually, we were expecting to see that that dominance was not that important for the kinds of theoretical reasons we talked about yesterday, but we found, the result of essentially 0 is even more extreme than we were anticipating. Yeah.
Michael Bennett looked it up. It's 3,952 read SNPs.
Fantastic. Thank you, Michael. Yeah.
Two questions. So one is if you could speak a little bit more about, you mentioned earlier that you added the X chromosome in this one.
Yes.
So if you could speak, and then secondly, do you now have enough power to control for cohorts? But cohort, like like if there is a cohort effect in it?
Yeah, so on the X chromosome, there were two questions. On the X chromosome, which I'm not showing here, we find some. There's a number of interesting results that relate to the X chromosome, which we looked at in both EA3 and EA4. The highlights are you get hits. It's about the number of hits that you would expect given the effective number of loci on the X chromosome relative to the autosomes. We find some inactive X activation in women for SNPs that are relevant for educational attainment. And it's just enough X inactivation such that the heritability of educational attainment due to the X chromosome is basically the same across men and women. Maybe that's the most interesting. There's also, on both the X chromosome and the autosomes, there's a very high genetic correlation. So you basically don't see sex differences in the, certainly on the autosomes. There's no sex differences on the SNP effects. The other question was about birth cohorts. So that's something we looked at in EA3, and we didn't revisit in EA4 because we were just expanding one of the cohorts. But in EA3, we looked at heterogeneity across cohorts in terms of SNP effects. So like, what is the genetic correlation when you go from one average cohort to another, some other cohort? And on average, we estimated about 0.7 for the genetic correlation. So it's not perfect. So there's some validity to that early criticism that you're adding noise by combining across cohorts. But it's high enough that on net, for cohorts of the size that we have, we're adding more signal than noise by adding cohorts and meta-analyzing. And we also look at some of the factors that contribute to higher heritability or higher genetic correlation between cohorts. So I encourage you to look at the supplementary materials for EA3 if you're interested in that. Yeah?
So following up on that question, first on the X chromosome, it's more common. There used to be in popular culture this conception that the intelligence comes from the mother because through older, I think it was linkage studies, they had found some X-linked intellectual disability.
Yeah.
And it was both sides of the X chromosome has a special role in heritability of intelligence. So just to make clear, you did not find that.
We found no evidence for a special role for the X chromosome in educational attainment, which is related to cognitive performance, but not quite the same thing.
Right, right. Second, was also her question regarding the controls and whether we should control for like family SES, certain type of socioeconomic index. You said one answer is that these indices are not available in every cohort. I was just wondering if there is a statistical reason, a methodological reason that perhaps we shouldn't control when we're doing Zin discovery for socioeconomic status. Is it because it could be a mediator and we don't want to control for it because of collider bias, maybe? So I know in econometrics there's this tendency to control for everything because you have omitted variable bias and you want to always have every relevant variable provided.
Well, so I disagree with that characterization of econometrics. There's a lot of concern about over-controlling, controlling for mediators. I mean, I think you're right that to the extent that socioeconomic status is a mediator, it would be a problem to potentially control for it. But I think for a person's educational attainment, their own socioeconomic status, I don't think is likely to be a mediator. It's more like their parents' socioeconomic status. So it might depend on exactly what measure you had about whether that was a concern of yours, if you're trying to get closer to a causal effect. But the more general point, I absolutely agree with. What you control for in the GWAS is going to affect the interpretation of the thing you're getting out. And I think one way to think about it is going back to that model that we started with in the very first lecture and think about what's in the set of control variables, and that's going to determine which best linear predictor we're talking about. It's best linear predictor controlling for something, which might be ruling out certain mechanisms or not ruling out mechanisms, or a causal effect or not a causal effect. And so there's a different interpretation for the coefficient depending on what those control variables are.
Okay. So in general, do you think it's a good idea in the future potentially to control for socioeconomic status?
I mean, if you're trying to get closer to the causal effect, I think controlling for parental socioeconomic status probably would be a good idea. But I think even better is just going to be to do within family GWAS, which is going to just control for all of the confounds and get you the causal effect. I mean, approximately. OK, so let me end. So summarizing the gene discovery lectures, beware which gene discovery research you rely on. Some of it is more reliable than others. The general finding for behavioral phenotypes is that individual loci have small effects. I've talked here about educational attainment as an extended example, but we have looked at other phenotypes, risk tolerance, subjective well-being, other things, and we virtually always find this. There are, we called this there's a paper, we called this the fourth law of behavior genetics, adding to Turkheimer's 3 laws. There are some exceptions, though. And so I want to make sure you come away with this lecture understanding that there are some cases of variants that have large effects. Here are probably the 3 most relevant for research you might be interested in. There's the FTO gene, where it's well tagged by a SNP that has a fairly large effect on BMI. There's one particular SNP that is colloquially called Mr. Big because it's the one with the big effect. And it has a relatively big effect on smoking. These effect sizes are on the order of like 1/3 of a percentage of R squared. So it's not huge, but it's large relative to what it's 10 times larger than we're seeing for effects on things like educational attainment. And then there's ApoE on Alzheimer's, which is a much bigger effect. That's like 15 or 20% of the variance. So you could study, if you're interested in some kinds of research designs require understanding the biological mechanism, and you're going to have a much better shot than focusing on individual SNPs if you need to understand the biological mechanism. And in order to have power, you're going to want to focus on SNPs with relatively big effects. So for some research designs, these could be useful. I think much more commonly in the social science applications, we're going to be interested in using polygenic indexes, which is going to be a major topic tomorrow. OK, and then finally, do power calculations. You now know how to do it. You've written software to do it. So there should be no barrier to doing it in your own work. And do well-powered analyses. So do the power calculations and then only do the research if it's adequately powered. Because if it's not, you're not going to learn anything. And at worst, you might even create false positives, which you don't want to do. All right, so we'll break now and resume at 11.